This conference will now be recorded.
Okay, good evening everyone. In the last class we discussed about the all protein structures. Okay, and now we are going for the topic protein sequencing strategies. And what is meant by sequencing first? I think all are you joined. Malanya, Shalini, Vichy. Okay, what is meant by sequencing, ma? First, sequencing means okay. Only three members are joined, na?
Argadeep, other students will join or not if they are absent? Argadeep, are you there? Argadeep, are you there? Just respond to me. So today we have Salini, Mamunya, and Vidushi. I think other students are absent. Okay, ma'am, you go ahead, ma'am. They will join in the middle, ma'am. Argadeep, are you listening me? Argadeep? Still Naga and Priyama Kumari and Darshana has to join. Okay. So Anaga and other students are absent. So today, so these much students will be there only for today's class, ma'am.
Okay, okay, ma. Okay, anyway, we'll continue the class. Okay, here we are going to the section of protein sequencing strategies. And here, what is meant by sequencing means for the determination of the components that are present in protein. That means here, proteins are made up of amino acid. And what are the types of amino acids that are present in the protein structure? And that we have to determine, and that is called as the protein sequencing strategy. DNA sequencing is there, protein sequencing is there. From the DNA, proteins are coming. That means here, from the sequence of the DNA, here we are determining the protein sequence, which proteins, which amino acids are coming from the DNA. Okay, and here in the proteins are also different amino acids are present, and that we have to determine. Okay, and that is called as the protein sequencing strategies. And here, normally the sequencing, these are determined by using the chemical methods or by enzymatic method. These two methods are only used for the determination of the any sequence or DNA, it may be DNA or it may be protein. Chemical methods and enzymatic methods. These are mainly used for the determination of the sequence. That means the composition of the proteins. And these are determined by using the chemical and enzymatic.
This
By
conference will
Edman.
now be recorded.
Edman then proposed the alternative approach for the determination of extended sequence of proteins or peptides or whole proteins. And this is nowadays, this one is using. Nowadays, this technique is used for the determination of amino acid sequence. Here, see the structure. Actually, I said the structure also. Protein structure normally start with the amino terminal end and ends with the carboxy terminal end. And the remaining that contains the in the middle, the intermediate amino acid components are present. And that means here it is mainly starting with the amino end terminus and ends with the carboxy terminal. Okay, and this is about the amino acid sequence. And how can we determine the protein sequence? The normal strategy that is required for the protein sequence. First, we have to determine the protein sequence strategy means first we have to learn the structure of proteins. Already we learned about the structure of proteins and what it contains. It contains different structural patterns. That is the primary structure, secondary structure, tertiary structure, and quaternary structure. Primary means there is a linear chain of amino acids, whereas the secondary means that consists of the alpha helices, that is the folded form of the primary ones, and that contains alpha helices and beta pleated chains. And that means in between the chains, peptide bonds are responsible for the formation of alpha helices and beta pleated. Next, three-dimensional. Three-dimensional is the more folded form of the primary structure, and it consists of mainly disulfide bonds and hydrogen. And next, quaternary structure. Quaternary, that is a folded form of the structure, that is a supercoiled structure, and that contains mainly motifs and other binding proteins. Okay, that causes the folding of the amino acids, the folding of the polypeptide chains. Okay, and that causes the quaternary structure. That is the normal protein structure. That means here we have to determine the sequence means and that structure must be degraded. Must be degraded. That means we have to cleave the protein into different fragments and then only we can determine the sequence of that particular peptide fragments. Okay, and that are the strategies that are included here. Okay, it consists of mainly 8 basic steps. It consists of mainly 8 basic steps. What are the steps? That means if the protein contains more than one polypeptide chain, that means your protein sequence coming from the quaternary structure. From the quaternary structure, we have to break the proteins into individual amino acids. That means the steps that are coming from the quaternary structure. Here, if the protein contains more than one polypeptide chain, then the chain should be separated. The chains ought to be separated and these ought to be purified. And that is the first step. And next coming to the tertiary structure. What it contains the tertiary structure? The tertiary structure mainly contains the intrachain disulfide bonds and interchain disulfide bonds. And these disulfide bonds, these ought to be clean. These ought to be clean. And here, if these disulfides are interchain, they may be interchain or intrachain. Interchain and intrachain. And if these disulfides are interchain linkages, then second step must precede than the step one. That means we have to do the first, second step. Second step. That is the separation of the polypeptide chain that cleaves the disulfide bonds. Okay, and next coming to the third step. Third step. What is the third step? We have to determine the amino acid. That means each polypeptide is separated here. Polypeptide is separated. That means we have to determine the amino acid composition of each polypeptide. Actually, the polypeptide starts with the N-terminal end and ends with the C-terminal end. That means we have to determine the N-terminal residues and C-terminal residues. These are to be identified in the fourth step. Next, in the fifth step, each polypeptide chain is cleaved into smaller fragments. That means we separated the different polypeptides and we have to determine the amino acid composition of each polypeptide. That means here each polypeptide chain should be cleaved into the smaller fragments. Smaller fragment. And then we can determine the sequence of that particular peptide fragments. Okay, that means it can be done by using the different chemicals or by enzymatic method. Okay, and next, the overall amino acid sequence of the protein can be reconstructed from the sequences in overlapping fragments. How can we determine the sequence? Means I will tell the process. How can we determine the sequence? How can we write the sequence after the cleaving of that polypeptide? Okay, and that is the determination of sequence. Next, then last but not least, here what is we have to determine? That means the bridges that are present in between the cysteine residues. We have to locate. That means the position of disulfide cross bridges that are formed between cysteine residues. These ought to be located. Okay, and this is the process and that we have to do for the determination of sequence. These are the 8 steps that mainly involving in determining the amino acid sequence of a protein. Okay, here in the fourth step, what we have to do? First, we have to determine the N-terminal and C-terminal. How we determine these ones? Means by using the different enzymes and by using the different chemicals. Okay, and that is the next method that is involving here. That is the N-terminal amino acid analysis. N-terminal, that is this protein chain, that is the peptide chain, that's starting with the N-terminal amino acid. That is the amino terminal. Okay, here it is to be reacted with a particular reagent that only labeling the terminal amino acid. That means the reagent should be added to the sample that only attaches with the terminal groups, terminal amino acid groups, terminal amino acid. That may be terminal means here amino present carboxyl bond, but here we have to specialize for the N-terminal amino acid. Okay, and then we have to hydrolyze that protein. Hydrolyzation, that is the next step. And next, we have to determine the amino acid by chromatography technique and comparison with the standards. And that means which amino acids are present in the particular sample, and that is determined by using a biophysical technique that is called as a chromatography technique. What is chromatography, ma? Chromatography means chromatography. Shalini, Vidushi, chromatography means anyone give me the response? Yeah, separation of molecules based on size, charge, affinity. Good, ma, Mavunya, good. Okay, here this chromatography, it should be compared with the standards. How can we compare the amino acids, unknown amino acids with the standards? What is the process that is involved here? Do you know about that? How can we determine the unknown amino acids by comparison with the standards? What is the process that is involved? Mavunya? Anyone? Shalini? Vidushi? Shalini, do you know? What complete sequence. Complete sequence. That means here, uh, that means we are using the enzymes. Enzymes means that giving the different components present in the mixture. Then we have to make the complete sequence by using the different fragments. Okay, that means here it is similar to like that of jigsaw puzzle. What is meant by jigsaw puzzle? I think you know about this jigsaw puzzle, and different cut pieces are present, and these have to be arranged to make the complete picture or complete the ones. Okay, and that is the puzzle. Okay, here also sequences are present. That means the composition containing the different amino acids are present. And that means here the complete polypeptide is determined by arranging these amino acids. Okay, and that means the arrangement of different amino acids that makes the protein, that makes the polypeptide. And that we have to determine by using a N-terminal amino acid and next proteolytic enzyme amino acids. Okay, and in 1955, Sanger was able to present a complete sequence of complete sequence of insulin. He determined the sequence of first insulin. Okay, and that is the determination steps that are included in Sanger's. You have to remember these all steps. Why means here in exam they will ask which steps are present, which one is absent, which are present. Why what is the first step? What is the second step? We have to arrange the steps also. Arrangement of steps, and these are also asked in exams. Okay, you have to clearly observe these patterns, how these are arranged, how we can determine the sequence. Okay, stepwise determination is necessary for sequence pattern. Okay, and next here in the first one hydrolysis there. Hydrolysis means hydrolysis mainly occurs by using the, here the peptide is hydrolyzed into its constituent amino acid by heating it with HCl, 6 normality HCl at 110 degrees for 24 hours. Here we have to determine the composition of proteins, means particular conditions are to be maintained for the denaturation of proteins. That gives only the particular amino acid, constituent amino acids are released by heating. That is the hydrolysis process. And this method is called as a Sanger's method. Mainly he used the reagent called as a 2,4-dinitrofluorobenzene. Fluorodinitrobenzene also. Okay, that is the main reagent. That is called as the Sanger's reagent. Okay, next. Okay, this is the Sanger's method. Here, what happened? 2,4-nitrofluorobenzene. First, it combines with the amino portion. This is the amino portion, and this one combines with the amino portion. Okay, and here it is coming this place, and that replaces the fluorine. That is the F molecules, and that gives a, okay, that gives a dinitrophenyl amino acid. Dinitrophenyl amino acid. And by using next after this here partial hydrolysis by different enzymes by proteolytic enzyme, and that gives a amino acid composition. And this is the protein sequencing by using the Sanger's method. And next coming to the Eggman degradation. Next one is the Eggman degradation. He mainly determined the ordered amino acid composition. Ordered amino acid composition mainly determined by Eggman degradation by using the degradation method. And by using this, next several sequences are came. Okay, automated Eggman sequences are now available for the determination of sequences up to 50 amino acids or long. Okay, and it is mainly based on that this Eggman degradation method. It is mainly based on the principle here. Single amino acids can be modified chemically. By using single amino acid, he determined the structure of polypeptide. Okay, here the single amino acids can be modified chemically. That means they can be cleaved from the chain without disrupting the bonds between the other residues. And that is the main principle here involved. What he done to only taking the single amino acids that can be modified by chemical method without disturbing the other residues, without disrupting the bonds between other residues. Okay, and here within minutes we can determine the sequence. Okay, and here usually amounts in the order of 10 to 100 picomoles will allow for successful completion. Okay, and this is the Edman method, and what he used, what is the process that is involved, what are the steps that are involved here in the determination of sequence by Edman method. Here it is mainly based on the reducing agents. Actually, disulfide bonds are broken by disulfide bonds are broken by mainly using the chemical 2-mercaptoethanol, while hydrogen bonds are urea. Urea mainly disrupts the hydrogen bonds. 2-mercaptoethanol disrupts the disulfide bonds. You have to remember that also, ma. Okay, here and also here during the process sometimes the reforming of the bonds occurs, reforming of the bonds occur, and that must be prevented by using the iodoacetic acid. Iodoacetic acid is mainly necessary to prevent the bonds that reforming the peptide chain. Okay, what is the process that is involved means here first we have to separate and purify the individual chains of protein complex. First we have to separate the individual chains. Then we have to determine the amino acid composition of each chain. First polypeptide breakdown, then giving the single peptide. Then that has to be disrupted, that giving the amino acid composition. Next we have to determine the terminal amino acid. First we have to determine the terminal amino acid sequence, and then break each chain into fragments under 50 amino acids length, and then separate and purify the fragments, then sequence determination. And this has to be repeated for the determination overall protein sequence. Okay, that means first certain length of the polypeptide chain is to be taken, and that is to be broken into chains, and then single chain containing the amino acid composition has to be determined. Next, in that first we have to determine the terminal amino acid composition. Then here the chain is to be fragmented onto 50 amino acid longs, and then the separation purification of fragments to be done. Then you have to determine the sequence of that particular fragment. These are the steps. And what is the method here mainly involves? What is the method here mainly involved? Here, first, the peptide fragments that are mainly digested by using the endopeptidases. Endopeptidases and exopeptidases are present. What are endo means? Endo means endopeptidases. What are exopeptidases? Anyone? Endopeptidases means? Okay, breaks the bond between. Okay, that means inner bonds out from inside. Okay, internal secretion. Yeah, yeah, ma. Here, endopeptidases. Peptidases means breaking the peptide bond. Endo means internal. Internal bonds are breaking. That means that first we have to digest the peptide bond fragments into different amino acids. That means peptide bonds are to be first broken, and that is mainly mediated by trypsin or pepsin. This is the enzymatic method. Edmonds method is the enzymatic method, ma. And also here chemical reagents are also used here. And here first digestion is done by using the endopeptidases, that may be trypsin or pepsin. Okay, and trypsin or pepsin, they digest the peptides at specific sites. I will tell what the specific sites are present here. Okay, and then by chemical reagents also, chemical reagents such as cyanogen bromide. Okay, different enzymes here giving different cleavage patterns. Different enzymes giving different cleavage patterns. Then the sequence is to be overlapped to construct the overall sequence. It is to be overlapped to construct an overall sequence. And that is the digestion into smaller peptide fragments. Okay, and what is the reaction that we have to determine the sequence means here the peptide which is to be sequenced, and that first it is to be absorbed onto a solid surface. Difference is there between absorption and adsorption. Absorption means only onto the surface, that is onto a solid surface only. And what is the solid surface here mainly used means one common substrate is glass fiber that is coated with the cationic polymers. Cationic polymers are used as the substrate. Cationic polymers. What are cation? Cation contains which charge, ma? Cations contains which charge? Cations. Yeah, positive charge. That means here normally amino acids exist in negatively charged. Here these polymers mainly attracting the amino acid that are of negatively charged. Okay, and next here the Edman reagent, main Edman reagent here used is phenyl isothiocyanate. Phenyl isothiocyanate is added to the adsorbed peptide, and it is to be added along with some basic buffer that containing 12% trimethylamine. And this reagent, it only reacts with the amine group of the N-terminal amino acid. N-terminal amino acid. That means here the terminal amino acid is recognized by using the reagent phenyl isothiocyanate. Phenyl isothiocyanate. That means here the terminal amino acid can be detached by using the addition of anhydrous acid. Anhydrous acid. Here anhydrous acid is mainly used for the separation of these N-terminal amino acid. That is we determine first N-terminal amino acid sequence. Okay. Okay, next. Here, this then it isomerases to give the substituted diphenyl thiohydroxide. Phenyl thiohydroxide, which can be washed off and it can be identified by chromatography. That means here the restriction digestion technique and PCR. And these all come in recombinant DNA technology. And here also DNA fingerprinting is there for the determination of DNA sequence. And like that out here, the protein sequence, it is determined by using the peptide mass fingerprinting or protein fingerprinting. Protein fingerprinting. Here, what is the process that is involved here is, here the unknown protein molecule, first it is to be cleaved into smaller fragment, and these are measured by using the mass spectrometric technique. Mass spectrometric technique. And also another technique is there, that is a tandem mass spectrometry. Tandem mass spectrometry, and that describes the partitioning of mass spectroscopy into separate steps where fragmentation occurs in between these steps. That means here in the first one, what is here we doing? Here in the first one, that is the peptide mass fingerprinting, that uses the technique of just cleaving of protein into smaller fragments. Whereas in tandem means, that means here the mass spectrometry, here in the first one also mass spectrometer is used for the determination, but it taking place into different steps. It taking place in different steps. That causes a fragmentation into different steps. And that are mainly used for the determination. Tandem sequences are there. Tandem in a DNA sequence, a tandem sequence. That means the breaking of the protein into different fragments, and that means the stepwise determination of sequences, and that causes the tandem mass spectrometry. Tandem mass spectrometry, and this is a two methods that are mainly used for that. Okay, here mainly it uses the method that is called as the digested the peptide. First we have to digest the peptide ones by using the enzymes, by protease enzymes, or different enzymes can be used. Okay, then ionization by molding. Molding means here matrix assisted laser desorption or ionization or electrospray method. Here, that means here this contains the amino acids contains the different ions. Okay, here first we have to ionize the amino acid composition, and that is to be done by using the molding, and that is called as a matrix assisted laser desorption. Why it is called as a matrix? Already I said different types of matrices, that is the cationic ones, anionic ones, that are mainly used for the determination of sequence. Here also the same technique here also applicable, but a little change is there. Okay, in instrumentation you will know about this technique also. Okay, then here these ionized molecules, these are introduced into the instrument called as the mass analyzer. And this mass analyzer, it determines the masses of these ionized molecules, ionized amino acids or the ionized peptides. And by using these masses, it is now possible to know the amino acid sequence of the protein. Amino acid sequence of protein. And this is the method that is involved here. First, the digestion of the peptide bonds, and next ionization. Next, ionization of molecules that are introduced into the instrument, and that causes the measurement of each ionized peptide. And next, by using this, we can determine the amino acid sequence of the protein. Okay, here actually what is ionization means, here mainly electron gun is used for the breaking of these amino acid into different ions, ionization process, and that is occurring in mass spectrometry for the determination of amino acid. For the determination of amino acid, that is called as the mass spectrometry. I think this is this is similar to like that of in gene transfer method, particle bombardment method is there. Like that also, here also electron gun and that means the electrons are mainly responsible for the ionization of the amino acid, and that are detected by using the instrument called as a mass spectrometry. Okay, and just I'm giving the only principle and the process we will discuss in instrumentation technique ma. Okay, in instrumentation technique it will come. Okay, next, and this is about the mass spectrometry. Okay, and next we are going to the conclusion here. What is the conclusion regarding this here? In the for the determination of protein sequence here, first it has to be broken down into peptides. That is a peptide fragment, single polypeptide chains. That means polypeptide is to be broken down into single chain, and that is the breaking of disulfide bonds should be occur. Then the protein should be cleaved into smaller peptides, and then separation of peptide, then sequencing of peptide, then alignment of peptide sequence. Okay, and what are the enzymes here we are using for the hydrolyzing of the peptides? What are the different enzymes? Okay, I think proteolytic enzymes means proteolytic. Proteo means protein. Lytic means lysis. Here, the proteins are digested by using the proteolytic enzymes. Mainly, two enzymes are mainly responsible for that. Endopeptidases are present, exopeptidases are. Endo means internal bonds are breaking. Exo means the terminal ends of the amino acids that are determined by using the exopeptidase activity. Okay, and what are the types of endopeptidases here? We are using different types of agents here used for the endopeptidases, and that are of mainly trypsin, chymotrypsin, pepsin, elastase, thermolysin. These are the main enzymes, and that are mainly used for the endopeptidase activity. Okay, here agents mainly trypsin. Trypsin. That means here different enzymes have different cleavage site action. They act at different sites in the amino acid sequence levels. Okay, trypsin. It mainly acts at the carboxyl side of lysine or origin. It mainly acts at the carboxyl site of lysine or origin. Whereas chymotrypsin, it acts at carboxyl side of aromatic amino acid residues. What are aromatic amino acids? Amino acids. Aromatic amino acids means here tyrosine, phenylalanine, tryptophan. These are the aromatic. Here chymotrypsin, and that causes the breakage at the carboxyl side of aromatic amino acid residue. Whereas pepsin, it breaks at the amino side of aromatic amino acid. Okay, and next elastase enzyme, it acts at carboxyl side of alanine, glycine, and serine. Alanine, glycine, and serine. Okay, and these activities done by elastase. Next, thermolysin. Thermolysin, it mainly acts at the amino side of amino acids like that containing the hydrophobic side chains like leucine, isoleucine, or phenylalanine. And these are the different amino acids, different enzymes, and that acts at the specific site of cleavage. That means by using these enzymes only, we can break the peptide bonds in the proteins, and that causing the releasing of different amino acids, and that causing the determination of amino acid sequence. Okay, and these are the different types of enzymes. You have to remember these enzymes, ma. It's very important. Trypsin, chymotrypsin, and pepsin. These three enzymes are most important. Trypsin, chymotrypsin, and pepsin. These are mostly important. Okay, and next coming to the exopeptidases. Exopeptidases. What are exopeptidases? Here, exopeptidases, that means these are acting at the outside of the chain. That means the terminal sides of the chains, and that are broken by exopeptidases. Here mainly carboxy region is present and amino terminal regions are present. Carboxypeptidases and aminopeptidases, and these are the main exopeptidases that are removing the terminal amino acid residues from carboxy and amino terminal of polypeptides. Okay, carboxypeptidases A mainly cleaves C-terminal peptide bond of all amino acid residue except proline, lysine, arginine. Except proline, lysine, arginine. Whereas carboxypeptidase B, it is effective only where arginine, lysine or the C-terminal residues. Well, arginine, lysine or the C-terminal residues, then carboxypeptidase B is effective. Okay, whereas carboxypeptidase C acts on any C-terminal residues. Any C-terminal residues. And these are the different types of enzymes and that are mainly responsible for the digestion. Okay. Okay, and this is all about the protein sequence determination, that mainly using the Sanger's method, advanced method. In advanced method, generally the chemical reagent is used, and also by changing the media, that is the pH conditions, we can determine the sequence. Okay, and here the digestion of the peptides, that is mainly done by using the different types of enzymes. And these are the different types of enzymes, and that causes the degradation of the proteins. Okay, is it clear, ma? All of you? Is it clear? Vidushi, Shalini, Priyama Kumari, any doubts, ma? Okay. Is it okay, all of you? Okay, ma. Next, we will go for the questions. Here, choose the mismatch here. The first question, what is the answer for this? Here, phenyl isothiocyanate, and that causes the hydrolysis of peptide bonds on the carboxyl side of lysine and arginine residue. Whereas B, diacetyl chloride, identification of amino terminal residue of peptide, chymotrypsin, hydrolysis of peptide bonds on carboxyl side of aromatic residues, CNBR, that is cyanogen bromide, and that causes the cleavage of peptide bonds on the carboxyl side of methionine. And which one is the incorrect option? Which one, ma? Phenyl isothiocyanate. What is phenyl isothiocyanate? It is Edman's reagent. Yeah, Edman's reagent. Edman's reagent, it mainly binds at the amino terminal end, right? That means here phenyl isothiocyanate, it is not causing the hydrolysis of peptide bonds on the carboxyl side of lysine and arginine residues. Okay, that means here the first option is the, that is a clearly mismatch